Assalamu alaikum everyone. Today I will be sharing with you two remaining drugs from the chapter of the antiviral drugs. First one is the ribavirin, second one is tromantatine hydrochloride. So let's start ribavirin. You can see in its chemical structure we can divide its structure into three parts. First one is this ring part. Second is this heterocyclic ring, and the third is this amine part. So what is this ring? It is called as the ribose. Okay, so if you remember, the ribose can be in the straight chain. Okay, ribose can be in the straight chain, or it can be in the cycle form. If you remember the carbohydrates, you will remember this ribofuranose. Why it is called furanose? It is a five-membered ring. That's why it is called furanose. But this, if this is in the if this cycle is in the six-membered ring, that will be called as the pyranose. So you can see in both are the structures of the ribose, having the same number of the carbon and the hydroxyl group, but due to the difference in the arrangement their name vary. This one is D-ribose and this is called as the beta D ribofuranose. Why beta D? Because various isomers are available. Similarly, in the ribavirin structure there is also a isomeric form which is present over here. Let's see in another structure of the ribofuranose. You can see the alpha beta bonding. Let's have its IUPAC name. This is the beta D furanose or 5 hydroxy. You can see the 5 carbon over here. This is the 5 carbon 5 hydroxymethyl oxalane. This ring is called as the oxalane. Okay, on the oxalane on 2 3 4 the hydroxyl group are present. You must be thinking why this is 2 3 4. When we are numbering on the basis of the oxalane, then this will be 1 2 3 4 and this will be 5. But if you are numbering on the basis of the ribose, then the numbering vary. But when we are numbering on the basis of the oxalane, then this is 1 2 3 4 5. So on the 5 this hydroxymethyl is present. Okay, and on the 2 3 4 the hydroxyl group are present. How many hydroxyl group are present? 1 2 and 3 hydroxyl group are present. That's why it is called as the triol. Okay, so this is the D-ribose, beta D-ribose. Let's see this structure. You can see it is a 5-membered ring with the 3 nitrogen and 2 double bond. Let's have a look on the structure. This is called as the triazole. We already discussed this azole as well as why it is called triazole, because 3 of the nitrogen are present. But these triazole may vary according to the position of the nitrogen. Either it is 1 2 4 triazole or 1 2 3 triazole. Okay, in our structure we have not 1 2 3, we have 1 2 4 triazole. Okay, now the last part is this amine part, or you can say this is the carbon, and it is called as the amide. Okay, amide, when the hydroxyl group of the carboxylic is replaced by the amino, it is called as the amide. Okay, amide group, simple amide or the substituted amide for your reminder. So its IUPAC name is 1 beta D ribofuranosyl on the 1 position. Again I am repeating, if it is the ribose, the numbering will be different. Okay, so on the first carbon there is the attachment of the triazole. Which triazole it is? it is 1,2,4-triazole. Okay, on the third position of the triazole. Okay, on the third position of the triazole, we have the amide group attached to it. Okay, so in the structure we can see that 1, 2, 3, 4, 5. So this 3 is the 3 of the this triazole ring, and 1, 2, 4, the nitrogen is present on the 3, carboxamide is present. Okay, so basically ribavirin is a compound which is the purine nucleoside analog. Okay, what is nucleoside? Nucleoside having the nitrogenous base as well as the sugar part binded to it. Okay, so it is not exactly the nucleoside, it is the nucleoside analog with the modified base, and the which base? Nitrogenous base, and a D-ribose sugar moiety. Okay, next let's have the definition of the furanose. What is furanose? A quick definition for it. Furanose is a collective term for the carbohydrates that have a chemical structure that include a 5-membered ring system consisting of the 4 carbons and 1 oxygen atom. The name derives from its similarity to the oxygen heterocyclic furan, but the furanose ring does not have the double bond. If you remember the structure of the furan, it have the same chemical structure but having the unsaturation, but this furanose do not have any double bond, so it is called as the furanose. Okay, let's have a look on the mechanism of action of the ribavirin. The mechanism of action of the ribavirin is not known completely, but the broad antiviral spectrum of the ribavirin, however, suggests multiple modes of action. Okay, so these are the suggestions. The it is not completely known. It is suggested that there are various pathways by which this ribavirin is working. The first one is the nucleoside is bioactivated. The nucleoside means ribavirin. The ribavirin is first bioactivated by viral and the host cellular kinase, means by the enzyme that are from the viral or the host, to give the monophosphate ribavirin and the triphosphate ribavirin. So ribavirin itself is not an active form. It is first converted into the monophosphate, and then the triphosphate, means the phosphate group is added to it, or the triphosphate is added to it, and then it performs the activity. Which activity? Ribavirin monophosphate inhibits inosine monophosphate dehydrogenase. Again, it is an enzyme which is important for the viral cycle. So it is inhibiting this enzyme inosine monophosphate, thereby preventing the conversion of the inosine monophosphate to the xanthine monophosphate. Xanthine monophosphate is required for the guanosine triphosphate synthesis. Okay, guanosine triphosphate synthesis. What is this? Guanosine triphosphate is a nucleotide. Okay, so ribavirin triphosphate inhibit the viral. This is the another suggested pathway, that ribavirin triphosphate inhibit the RNA polymerases. Okay, it also prevent the end capping of the viral messenger RNA. Okay, so during the replication of the RNA, obviously the nucleotides and the nucleosides are required to make up their DNA or RNA. So the ribavirin is preventing either the synthesis of the messenger RNA, or either it is inhibiting the guanosine triphosphate or the combination of the nucleotides so that it can prevent its further replication. Okay, now let's have a look on the general properties of the ribavirin. Ribavirin occurs as the white crystalline polymorphic solid. It is soluble in water and chemical. So let's have a look on the indications of the ribavirin. Ribavirin inhibits the replication of the wide variety of the RNA and DNA viruses, including orthomyxoviruses, paramyxoviruses, arenaviruses, Bunya viruses, herpes viruses, adenoviruses, pox virus, vaccinia, influenza virus type A and B, parainfluenza virus, and the rhinoviruses. It means that it have the activity against many viruses, but ribavirin is only indicated or only used for the one therapeutic condition. Okay, it is indicated for only one therapeutic condition, that is it is used in the treatment of the severe lower respiratory tract infection caused by the RSV. What is RSV? RSV is the respiratory syncytial virus, and it is basically in the carefully selected hospitalized infants and young children. It is this RSV is also called as the human respiratory syncytial virus and human orthopneumovirus, but basically it is more common in the infants and young children. It is also reported that it takes place in the children below 2 years, and it is very common in young children's. Okay, so it is basically a very common, and the contagious means can be transferred to other person or the child, that causes infection of the respiratory tract, which lower respiratory tract. And the symptoms are cold like, same like cough, runny nose, or the sore throat or fever, and it may last for 1 to 2 weeks. So it is only indicated for this, ribavirin is indicated or approved for this condition, RSV. But the unlabeled use of the ribavirin include aerosol treatment of the influenza type A and B, and oral treatment of the herpes, genital herpes, and the Lassa fever. Lassa fever is acute and the viral disease caused by the type of the rat, but it is not common over here. It is common in the West Africa. It can be life-threatening. Why? Because it is a hemorrhagic virus. Hemorrhagic virus means that it may cause bleeding. And although 8 out of every 10 people with the viruses have no symptoms, but it can affect the liver, kidney, or spleen. That's why it can be fatal. Ribavirin does not protect the cells against the cytotoxic effect of the AIDS virus. Means it can inhibit the virus, but it cannot inhibit the cytotoxic effect of the virus. Pharmacokinetics of the ribavirin. It is rapidly and extensively absorbed following the oral absorption, metabolized by two pathways. Okay, so the first pathway is the reversible phosphorylation, as we discussed in the mechanism of action, that the ribavirin is first converted into the monophosphate form, ribavirin monophosphate. It means to the ribavirin one of the phosphate group is added, or the 3-phosphate group are added, so it is converted into ribavirin triphosphate. So during metabolism, there is reversible phosphorylation, means there is removal of the phosphate group. Second pathway is the degradative pathway, involving deribosylation and the amide hydrolysis. In the second pathway, either this ribose part is removed or this amide part is removed. Toxicity of the ribavirin. it may cause anemia, headache, abdominal pain, and lethargy have been reported in the patient receiving the oral ribavirin. Okay, in the previous slide we have discussed that the it can be given in the aerosol form. So when it is given in the aerosol form, it can cause the deterioration in the respiratory function, bacterial pneumonia, pneumothorax, and apnea have been reported in the severely ill infants and the children with the respiratory syncytial virus infections. Okay, next is the tromantadine hydrochloride. You can see in the structure of the tromantadine, we can break down its structure for understanding into various parts. So let's see this part. This they have no any unsaturation, and it is a tricyclic. If you see closely, I will mark these cyclics separately. This is the one cycle, okay, and this is another cycle, okay, and this is the third cycle. I was coloring it to show you the different parts of this tormented this ring. What this ring is called? This ring is, you can see the numbering of it. It is called as the adamantane. Okay, what is adamantane? Adamantane molecule can be described as the fusion of three cyclohexane ring. Okay, it is a member of the polycyclic alkane. So three cyclohexane ring are three rings are fused together and they make the 10-membered polycyclic alkane. It is also the member of the polycyclic alkane. Okay, and it is also called as the tricyclodecane. Why decane? You can see the number of the carbon which are present in the structure are 10. 1 2 3 4 5 6 7 8 9 10. This over here is basically connected this in this structure, just showing you that the bonding is crossing from here. So basically this is a three-dimensional structure, but if we draw it in the one dimension, it will look like that. How you can draw this? First you will draw this uh, cyclohexane, okay, then you will, from this point, from the one point, you will draw like this down, and then this, and you will connect it on the 3. You can see it is connected on the 3 and 1, and lastly you will start from the 7th and okay, sorry, let me do it again, connected on the 5th. Okay, so this is the adamantane. Let's see the other parts. Over here we have the amide part, okay, acetamide. This with the 2 carbon oxygen is there. We can also call it, the 2 carbons are there, we can also call as a ethoxy, and on this nitrogen 2 methyl groups are there. So we have the dimethyl. So again, if we give its IUPAC name on the nitrogen, this adamantane is attached, and if we give name on the basis of the acetamide, so on the acetamide the adamantane is attached, as well as the ethoxy attached, as well as the dimethyl amino is attached. So the in the IUPAC name, the furthest are named first. Let's see its IUPAC name. N, on the N, on this N, one adamantile, you can see adamantile is attached on the 1 position. Okay, so this is one of that, one adamantile. 2 is of, this 2 is of acetamide. Okay, so 1 2 2, we give the big bracket Y, because there are two attachments are there. This attachment and this attachment is there. Okay, two attachments are there. So on this, I told you this 2 is of the acetamide. Now about this 2 is of the ethoxy. So 1, 2. On the second, we have this dimethylamino ethoxy acetamide and hydrochloride. Okay, let's have a look on another IUPAC name. 1. This name is on the basis of the adamantane. Okay, over here you will see admantyl because it is binded to the acetamide. Over here we are giving name on the we are saying that on the adamantane these attachments are there. So on the adamantane, on the one position dimethylamino ethoxy acetamido adamantane. Okay. Tromantadine hydrochloride is a hydrochloride salt form of the tromantadine, which is tromantadine is the cyclic amine. Okay, so separately it is an antiviral, but hydrochloride means it is a salt form for the better activity of the tromantadine. Okay, mechanism of action of the tromantadine hydrochloride. It changes the glycoproteins of the host cell, thereby preventing the entry of the virus. So these glycoproteins are present in the cell wall of the host. So why these are changes by the tromantadine hydrochloride? So that virus cannot enter into the host cell and replicate and produce the symptoms. It also inhibit the absorption of the virons to the cell surfaces, as well as the penetration and uncoating of the virus. This statement is again repeating that it is inhibiting the entry of the virus or the penetration of the virus, as well as the uncoating of the virus. What is virons? Viron is basically a term given to the entire virus particle consisting of the outer shell called the capsid and the inner core of the nucleic acid. So we can say virus as well as the viron can be used as a synonym. What about so we discussed that the this drug tromantadine hydrochloride changes the glycoprotein host cell so that it cannot be absorbed in the host cell, or it prevent the penetration into the host cell, as well as it prevent the uncoating of the virus. What is uncoating? Uncoating is the process by which an incoming virus is disassembles in the host cell to release the replication component replication competent viral genome. So basically to replicate, the virus need to disassemble first, so that it opens up and give and release the replication competent viral genome. Indication of the tromantadine hydrochloride: it is used against the herpes simplex virus, the condition which is caused by these viruses, and it can be caused in the allergic dermatitis, the inflammatory condition caused by the any allergic reaction. Toxicity reaction is the allergic skin reaction. So this is all about the antiviral drugs, and on this our chapter is completed, and inshallah in the next lecture we will be discussing the immunosuppressive agents.